que además de un intenso currículum en los últimos años, casi sorprendente la capacidad de trabajo de esta joven investigadora que viene dedicándose no solo a la atrofia de nervio óptico de Leber, sino también a otras enfermedades mitocondriales. Pero más allá de todo esto, quiero agradecerle que haya vuelto después de la que le dimos en el último encuentro en Sevilla, en la que explotamos al profesor Carelli y a ella misma, y quiero también aprovechar para agradecerle la absoluta disponibilidad de Chiara, todas a 24 horas del día, estando en Italia, estando en Estados Unidos, estando en congresos, lo cual no se pueden imaginar lo importante que es para una asociación. No es solo su investigación, que lo es, no es solo su simpatía, que también lo es, sino sobre todo la disponibilidad que tiene para con esta asociación. Con lo cual, les dejo, muchísimas gracias y esperemos seguir manteniendo los tiempos.
Thank you, Pedro, for the beautiful words. I'm very happy to be here today. My talk will be focused on the possible occurrence of plus features in labors. I will talk about the possible occurrence in Leber's hereditary optic neuropathy of migraine, some movement disorders, peripheral neuropathy, MS-like disorder, hair disorders, hearing loss, and other features. In 2003, Cupini and co-authors described this big family carrying the 14484 mutation, and the authors noticed an higher recurrence of individuals with migraine features. The occurrence of migraine was existent both in carriers of the mutation and Leber's individuals. And the authors at this time speculated that some connection between migraine and Leber's can be found. Previous studies demonstrated that migraine itself has a mitochondrial basis, possibly. And this study in particular demonstrated by magnetic resonance spectroscopy the occurrence of mitochondrial dysfunction in migraine. The connection between migraine and mitochondria can be also underlined by the efficacy of the use of riboflavin, vitamin B2, in Leber's patients suffering migraine. The rationale for using this drug is that this drug is a precursor of a flavin, which helps the mitochondrial enzyme respiratory chain activity, in particular complex 1 activity. So our suggestion is to use for patients, for Leber's patients suffering migraine or migraine with aura, riboflavin 400 milligrams per day as a prophylactic agent when the recurrence of migraine attacks is over 2 attacks per month. And in this interesting study, Di Lorenzo and co-authors in 2009 demonstrated that possibly the response to this drug is influenced by the mtDNA haplogroup. So they observed that people carrying the haplogroup H usually do not respond to riboflavin. Instead, the people with the non-H haplogroup, they have a good response. And this can be related to the fact that the H haplogroup itself has an increased activity of complex 1, and so the effect of riboflavin can be less. The connection between these two entities, migraine and mitochondrial, is also underlined by the fact that many mitochondrial diseases, in particular MILAS patients, suffer migraine in a very high frequency. And what we found in this family in 2009 is the high occurrence of migraine features also in patients not carrying the 3243 mutation. This means that probably the recurrence of migraine is correlated to the maternal line and so probably related to mitochondrial dysfunction itself. The possible genetic influence on mitochondrial DNA on migraine is still under investigation, and now the possible role of mtDNA methylation is under study. So we collected our data of the possible occurrence of migraine in our patients. We interviewed by a structured interview 100 subjects, 27 affected Leber's patients, and also their relatives along the maternal line. What we found is an high prevalence of migraine if compared with the prevalence expected in the general population. So what we can say is that the risk of suffering migraine in Leber's patients is higher in Leber's families. And this does not depend on the fact of being or not affected, but is related to the presence of the mutation. We did not find any difference in terms of mutation, so the risk it seems the same for the 3 Leber's mutation, and this our data confirms the fact that migraine can be related to the presence of Leber's mutations and the possible role of mitochondrial dysfunction in the pathogenesis of migraine. Another possible feature that we can observe in these patients, even though it's very rare, is the occurrence of myoclonus. In 2008, we described the 2 families, Leber's, one 11778 and one 3460, which showed the occurrence of myoclonus at neurological examination. This myoclonus was cortical myoclonus, and we demonstrated the presence of a spike before the occurrence of myoclonus by means of back averaging. And the possible finding of this neurological feature, we speculated that can be related on the occurrence in these particular families of other rare mtDNA variants that possibly modulate the phenotype. This means that usually we do not observe this feature in our Leber's patients, but if in particular families we have other mtDNA variants, they can modulate the phenotype. Another possible, even though very rare neurological feature in these families is spastic dystonia, which is reported only in association with the rare 14459 mutation. It is not described with the other classic mutation, and is characterized by the occurrence of these characteristics lesion in the basal ganglia in the brain, which leads to a neurological feature characterized by dystonia. Even though in the same family you can find patients with only Leber's and patients with spastic dystonia as well. The description of this entity is started in 1986, this observation, and the association with this particular mutation was then confirmed. Here I'm just reporting one example, one of our family carrying this particular mutation. So you can find that this individual who presents the phenotype with spastic dystonia. In the same family we have this individual who is affected only by hereditary optic neuropathy. And the brain of this patient showed these characteristic basal ganglia lesions. Another feature which is very controversial in the literature because there are scattered reports but never systematically investigated is the possible occurrence of peripheral neuropathy in these patients. Our impression is that patients usually do not complain symptoms suggesting the occurrence of peripheral neuropathy, and also neurological examination did not show any features suggesting this entity. But if we look at post-mortem tissue, in this case was the brachial plexus from a patient suffering labors, you can see some degenerative features compared to control in this particular case. And if you look at the single fibers, you see in labors a large amount of degenerated fibers also in the peripheral nerve. I'm now moving to the MS-like, or also known as Harding's disease, the possible occurrence of MS features in labors. This is a very controversial entity. The description of the possible occurrence of multiple sclerosis-like illness in women carrying the Leber's mutation was made by Anita Harding in 1992. And she observed the presence of Leber's patients carrying the Leber's mutation with features resembling multiple sclerosis. This recent study in 2013, which was run by the English group, was based on a retrospective review of all Leber's cases with MS, and also a prospective study revealing all the prevalent cases showing both MS and Leber's. And the conclusions of the authors is that although the co-occurrence of MS and Leber's is likely to be due to a chance, so the prevalence of the MS that these authors found was the same that you should expect in the general population. So the conclusion is that is not a causal relationship, but it's a causal relationship. Even though this is the conclusion of the paper, the authors also noticed that the features of MS in these patients are different from those that we usually observe in MS, and so they underline that the clinical features of Leber's plus MS are atypical for Leber's for these particular clinical features, which is the occurrence of multiple episodes of vision loss, long interval between eyes, and the predominance of women, which is unusual for Leber's, and atypical for MS, the absence of ocular pain when the patient loses vision, and also the poor visual prognosis. This is just the summary of the clinical features of this population. And if we look at the MRI of these patients suffering both MS and Leber's, we found that the appearance of white matter lesion is the same that we usually observe in MS itself. And female Leber's patients are at higher risk of presenting this kind of lesion, even though they are not always symptomatic. In 2015, we published this paper looking by advanced MRI technique, which is diffusion tensor imaging, at the brain white matter pathology in mitochondrial optic neuropathies, and we found that both Leber's and especially OPA1 patients displayed abnormal white matter changes, which is in Leber's limited to optic radiation and in less extent to acoustic radiations, is more widespread in patients carrying OPA1 mutations. The possible link between myelin and mitochondria is still controversial, and there are nowadays evidence that at the basis of MS there is mitochondrial dysfunction, or mitochondrial dysfunction can play a role in pathogenesis of MS. At the same time, mitochondrial mitochondrial mutation can have a role in trigger autoimmune response in predisposed individuals. So possibly the two entities, Leber's and MS, are modulating reciprocally themselves. This is just an example of one of our patients carrying the 11778 mutation who displayed brain features resembling MS. We now move to the possible occurrence of heart problems in Leber's. In the past, there were some reports pointing to the possible occurrence of Wolff-Parkinson-White and pre-excitation syndrome in Leber's. Although there are some reports suggesting the occurrence of this entity in Leber's, there are not systematic studies investigating specifically this issue in large cohorts of patients. Another observation is the possible occurrence of left ventricular hypertrophy in Leber's and some conduction problems, defects in these patients. In general, we can say that we do not observe cardiac feature in Leber's based on our clinical experience, but we recommend to perform at least once EKG, cardiac exam, one echocardiogram, and in some selected case, especially if patients are symptomatic for syncope, loss of consciousness, or palpitation, we also suggest to perform the 24 hours Holter EKG. Another feature which can be observed in Leber's is the presence of hearing problems. Also, this is a controversial finding because if you look just at the auditory function in this patient, usually patients do not complain auditory problems, but if you use more sophisticated electrophysiological testing, you can find some abnormalities. But auditory perception in general is maintained, as also demonstrated by this study from Patrick Huaman and co-authors. So our suggestion is to perform an audiometry if the patient complains auditory problems, but usually is not a problem that somehow interfere with the quality of life. And lastly, I just want to mention the possible occurrence of very, very rare presentation in Leber's, and I want to underline that this presentation occur usually only with rare mutation, not with the classical Leber's mutation, which accounts for the 90% of cases. And these presentations usually are in association with the rare mtDNA mutation. We described, for example, this young boy with the rare 4171 mutation in ND1, who presented with the acute onset of vomiting and vertigo, and a brain MRI showed this lesion in the brainstem, which resemble somehow the presentation of Lee-like syndrome. This lesion, over the course of the year, they completely disappeared, so the prognosis in this case was very good. And we described another case with another rare mutation, which is the 3890 mutation in ND1. And also in this patient, we demonstrated the presence of bilateral brainstem lesions, which disappeared during the course of the disease. So the message here is that these unusual presentations are very rare, and they are described usually with the very rare mtDNA mutation. Another possible phenotype that we can observe only with this rare mutation is the presence of MELAS features, which is a disorder characterized by the occurrence of stroke-like deafness, myopathy, and lactic acidosis. And there are some reports pointing to this possible co-occurrence of MELAS in labors. Here you see there is a long list of possible mutations in mtDNA, and these are the possible phenotypes associated. You can see that the MELAS overlapping phenotype usually occur with this very rare mutation. So the message is that if the clinical features of the patients are suggestive of labors, we should, and we do not find the classical 3 mutation, we should screen the entire mtDNA for the possible occurrence of other rare mutations, which account for the residual 5–10% of cases. I would like to thank all the collaborators and you for your attention. Thank you.
Muchas gracias a la profesora Chiara La Morgia. Los rumores en las salas mientras iba exponiendo la comunicación pues dan a entender que muchas familias se han visto identificadas en algunos de los casos que se han planteado, incluso en el día a día con los socios, y es cierto que se dan muchos de estos, con lo cual creo que hay lugar para la investigación y espero convencerlos de que cuenten con nosotros para ello. De todas formas, el tiempo apremia, le agradezco que seguimos en hora y continuamos con uno de los grandes especialistas en la enfermedad de LHON. Desde los años 80, desde su tesis de laurea, lleva trabajando sobre esta enfermedad y así continuó en sus cursos de doctorado, en sus estancias de investigación, en sus proyectos, con lo cual es un referente ineludible en el mundo europeo para la investigación de LHON y lo que para el mundo universitario es también destacable, ya maestro de nuevas generaciones. Con lo cual, hablar del currículum del profesor Carelli sería muy largo y no es lugar para hacerlo. Pero tengo que terminar diciendo también el mismo agradecimiento, extender el mismo agradecimiento que he dicho para la profesora Chiara La Morgia. Desde que nos pusimos en contacto con él, ha puesto muchísimas facilidades, nos guía como asociación en la parte científica, con lo cual no solo es un invitado de excepción, sino sobre todo un asesor científico, está en la recámara y siempre dispuesto para facilitarnos las últimas investigaciones y el camino por donde podemos seguir. Así que muchísimas gracias, gracias mille, el profesor Carelli, y dejarle sin más con su ponencia. Gracias.
Thank you, Pedro, and thank you, Asanol, and all of you for being here. I had the opportunity to see many faces that I already met in Seville, the last time we came to Spain. And I will try to give you some simple messages from very complicated slides. And I'll try to show you how much complicated is actually Leber's disease. So Chiara already showed you this long list of mutations, and by the way, I want to reassure everybody the occurrence of extraocular symptoms in labors is extremely rare, and in most cases is associated with rare mutations. None of the three is actually typically associated with these extra neurological features. Anyways, 90% of people, and many of you here, have one of 3 mutations. We already said the numbers for these mutations. I want to remind that what we know after 20 and more years of studies in cells derived from the patients and in cell models is that if this particular enzyme of the working in the mitochondria where the mutations are, which is complex 1, if this enzyme is not working properly, we can expect less energy production, we can expect more reactive oxygen species, which means more toxic byproducts, and both things together make the cells more fragile, and as has been said initially, the retinal ganglion cells, the retinal cells originating the optic nerve, are particularly sensible to these dysfunctions. We already heard, and you know, about the clinical features of labors, but there are 2 things that we are still facing as mysterious features in this disease. Why males are more affected, are more prone to get the disease, and why within a family there are many individuals carrying the mutation but not having the disease. And my belief, and our group of investigators is focused on this, is that the 2 questions are strictly related one to the other. So while only males, or the majority of patients are males, and while some people is actually not being affected. So the first place to see for a disease which is prevalent in males is obviously chromosome X, because males have only 1 copy of chromosome X, while females has 2. So if a mutation or a variant is in chromosome X, the males will express that variant in terms of pathology, while females can still use the 2nd chromosome X to compensate for that. Here the point is, without getting into technical issues, is that we all looked at chromosome X genetically, but up to date there is no consistent reproducible result showing that in chromosome X you actually have a factor, a genetic factor, in addition to the Leber's mutation in the mitochondrial DNA. What we thought a few years ago is that females and males are very different from the point of view of hormones. Females produce in a large excess compared to males estrogens, and they have cycles. And so we thought of using estrogens in vitro in cultured cells to see if estrogens were able to compensate for the mitochondrial dysfunction of these cells. These are cells generated from Leber's patients. And to make the story short, I'm not going to describe the single graphs that are illustrated here, but estrogens are able to make more mitochondria, to increase the number of mitochondria, and this compensatory response itself is good. So the dysfunction of mitochondria is partially improved by estrogens by increasing the number of mitochondria. This is an extremely simplified version. Obviously, biochemically and in the cell biology details, this is a little bit more complicated. However, this is the message. Essentially, all the dysfunctions that we can describe in cells derived from Leber's patients can be slightly improved by exposing the cells to estrogens. And this could be the reason why, essentially, most of the females can live with the mutation without developing the disease. But this could be a more general mechanism. And so that's why, being inspired by this work, we went to check if actually making more mitochondria is a general feature that regulates penetrance, which means if being able, either male or female, to react to the presence of the mutation by increasing the number of mitochondria could be actually the key to understand who will get the disease or not. So we had a few people working for many years on this project, and the main person is our collaborator in Rome, Carla Giordano, and then a few key persons in the laboratory in Bologna. And the only thing that I want you to look, it's a graph, but it's a simple graph. If you measure the number of mitochondrial DNAs, you heard this morning that each cell has many mitochondrial DNAs, okay? If you take the cells from the blood and you measure the number of mitochondrial DNAs in blood from the normal people, you have about 200 copies. If you get people with Leber's mutation who got the disease affected with optic neuropathy, then the number of copies is within 400. But if you get people having the mutation, relatives having the mutations without being affected, so without the disease, then the number of copies on average is definitely larger. So it's over 400 within 600. So this means that if you, for any reason that we still do not know, are able to react to the presence of the mutation, increasing sufficiently the number of mitochondria, you are probably safe and you will not get the disease. If you're not so good in doing this, you're risking more. And obviously, if you're male, you are risking even more because you don't have estrogens. If something is not clear, we will discuss later this. So here the conclusions are that increasing mitochondrial biogenesis, so being able to produce more mitochondria, is a key feature that distinguishes who, having the mutation, is remaining safe and healthy compared to who has the mutation and unfortunately gets the disease. But the most important here conclusion is that if we are able, with pharmacologically, with the drug, to increase the number of mitochondria in people carrying the mutation, maybe this will be a therapy to avoid the disease for prevention. So now I want to show you how environment can actually play a role on this mechanism that I just explained. And to do this, I have to remind that with my American boss, Alfredo Sadun, who unfortunately could not come to this meeting, he was invited, but he's a very busy man, we have been following for more than 10 years a gigantic family with Lebers in Brazil. This family is actually coming from a woman who immigrated in Brazil from Italy, and we have been able to find also the Italian part of the family. Please note that the Italian side of the family is about 45 people, and the woman immigrating in Brazil generated a 360 individuals family. So people in Brazil is happier than in Italy. So if we look at the single families of this very large pedigree, here you have the families where the mother is with black symbol, which means this woman is affected, and the black symbols in the kid means the number of people having the disease. So essentially, if you are a kid, a son of an affected mother, in 60% of males you have the disease, and in 30% of females. But then if you look at the other nuclear families where the mother is not having the disease but has the mutation, so the symbol is open, is not black, then you have an extremely reduced number of people having the disease in the next generation. So if you are son of a non-affected mother carrying the mutation, 40% of males can get the disease. No females get the disease. So this, in our interpretation, means that there is some other genetic factor cooperating with the mutation in mitochondrial DNA, regulating the penetrance, so the number of people getting the disease. But there is also a confounding factor deriving from the exposure to environmental triggers. And we now know from a very nice study in UK that tobacco smoking is extremely important. So I'll show you here 3 cases. These are 3 brothers from the Brazilian family. The first person got affected at 27 years of age and had a very severe classical Leber's disease. The other 2 brothers actually got the disease after 40 years of age, and they had a very mild form of the disease. Very mild. All 3 of them have been smoking and drinking their whole life in the same way. So this means, first, that if you smoke and drink, but particularly if you smoke, you get the disease. But then, if you get the disease early, probably is because of genetics, and if you get the disease late, probably is because you're smoking for too long. And then we looked at the paper that has been published by Patrick Chinnery and Patrick Uwaiman, and surprisingly, these people is a curve showing how people is over age, get the disease if you are not smoker. So obviously most of the people are getting the disease in the first 2-3 decades of life. If you are a slight smoker, so if you smoke for a short number of years, you're slightly worse. If you smoke a lot for many years, it looks like you're protected because you get the disease only after 40 years of age. Obviously, this is not a protection. The interpretation is different. The interpretation is, follow me very carefully, that these people probably would not get the disease. So the line here would be straight until the end, until the death, without disease, unless you're smoking for 40 years in a row. So these people are naturally protected from the disease, but because they have been smoking for a long time, at some point later in life, they get the disease. So this was the UK, the United Kingdom study, and we did the same thing in our families in Italy. And this is one of the rare cases where in science you are able to reproduce exactly in the same way the same result. So these people are non-smokers. They lose, they get the disease, they lose their sight within the first decade. If you're smoking for a short number of years, you're doing worse. But if you're a heavy smoker for 40 years in a row, you just get the disease later in life. So this is the peak of age of onset of the people which are not smokers, and this is the peak of age of onset of people smoking. So here we are facing the following situation. This is my interpretation. Maybe Patrick Chinnery and Patrick Uwaiman are not perfectly okay with this, but my opinion is that you have classical Leber's disease because the prevalent reason for having the disease is genetics. Mitochondrial DNA mutation plus something else, other mutations, which we do not know. But then you have a second group of affected people, Leber's mutation, you are protected, you would go without the disease unless you smoke for 40 years, and then you get the disease later in life, after 40 years of age. And I rapidly show you, just look here, these are the smokers, these are the carriers, people with the mutation without, these are the affected, people with the disease, these are the carriers, people with the mutation without the disease. These are non-smokers, these are smokers, these are non-smokers, and these are smokers. So this is the amount of mitochondrial DNA copy number. It's a little bit complicated, but here the observation is that if you smoke, you decrease the number of mitochondria. So the compensatory response, increasing the number of mitochondria, which we showed before, was a good thing to do. If you smoke, you actually do the opposite thing. And this is why you are getting the disease probably. So the recommendation is don't smoke. And as we did with estrogens, we also challenged the cells in vitro with smoking tobacco, and we showed that the number of copy of mitochondrial DNA is decreasing if you expose the cells to smoke. So we applied different complicated approaches. Some of these are actually has been introduced by the first speaker to search for these extra genes. We generated a long list of candidates. I'm not getting into the details. These are the most interesting genes, but the point is we need to validate if these extra genes, variants in these extra genes, are actually influencing the mitochondrial DNA mutation to promote the disease. And this is a work in progress that we are doing in collaboration with the people in United Kingdom. So nowadays, all the pharmaceutical companies look at labors as a model for doing trials. This is because labors is a disease which now is well known. You know what to expect from the disease, and so you know what you have to measure in order to demonstrate that the drug is actually working. And our favorite approach is, as a consequence of our studies, is to try to increase the number of mitochondria in patients with drugs. And the last few slides are dedicated to the fact that there are good evidences from the scientific literature that if you donate these groups called NAD, you activate the genesis of mitochondria. This is a very complicated, just skip it. But we did some experiments in vitro, and the higher lines indicate that cells are reacting well when you push the mitochondrial biogenesis. So you give drugs, the number of mitochondria go up, and the cells is happier. And we just published the fact that using phytoestrogens, which is a sort of estrogens without the side effect of estrogens. Obviously, you cannot give estrogens to males because you transform males in females. So it's obvious that you cannot use that as a therapy. But if you use this, for many reasons, these are able to do the same thing as estrogens and are possible to be used in humans. The names of these phytoestrogens are genistein, dazein, and equal. We used a combination of 3 of them. And the results of the combination of 3 of them is exactly the result that we got with estrogens. So this is a similar benefit. You increase the number of mitochondria. The red dots are mitochondria, so you go from here to here. So this is good. We can skip this. You lower the reactive oxygen species. So red is when you have the reactive oxygen species. If you have no red, means that the toxic byproducts of mitochondrial mutation, Leber's mitochondrial mutation, are disappearing. Okay. So this is my last slide. The group in Rome has been extremely helpful in all these studies that I have shown here today. This is the people in my lab. Some of you can recognize Chiara. And then we have other people working with us. And obviously, these are the funding bodies. Most of them are people, are association of mitochondrial patients. And then we have many collaborators. Patrick Chinnery and Patrick Uwaiman are extremely important now for the studies we are running in genetics, and Alfredo Saduni in the United States, my boss in the United States, is a very good collaborator of us even nowadays. Thank you for your attention.
Muchas gracias al profesor Carelli. Entiendo que es mucha información para ir haciendo un poco la digestión de ya las 4 ponencias que tenemos. Vamos a hacer otro receso y intentamos ser al menos tan puntuales como en la ocasión anterior para ya terminar con las últimas 2 conferencias. Gracias.